Hello, everyone, and welcome to part two of our screencast lecture on the urinary system. You'll recall whenever we left off last time, we were talking about nephron physiology. In other words, we were discussing how uh, the nephrons, through their activity, control the composition and concentration of the blood plasma and tissue fluid. And we said this was done through the formation of urine. Now, you'll also recall that the formation of urine involved three distinct processes we mentioned. First of these processes was filtration. During filtration, recall a filtrate of blood is going to leave the kidney capillaries, more specifically the glomerular capillaries, and the filtrate then, whenever it leaves the capillaries, will then move into Bowman's capsule. So this was a passive process because it was driven by slightly higher blood pressures that occurred in the renal corpuscle. The second component of urine formation, which is where we're going to begin today, is referred to as reabsorption. So filtration is a pretty indiscriminate process. Anything that's small enough to pass through the capillary beds, the glomerular capillary beds, and into the filtrate will be moved into the Bowman's capsule. This means there's going to be nutrients, there's going to be water, and there's going to be essential ions, as well as waste products that are going to move into the Bowman's capsule and eventually into the tubular part of the nephron. Well, we can't afford to get rid of the nutrients and most of the water and the essential ions. So they're going to have to be reclaimed, and this is done in the process of reabsorption. And the final component of urine formation of nephron physiology would be secretion. Now, secretion is an active process that removes additional undesirable molecules, let's say, like, for example, excess hydrogen ions from the blood that haven't been removed through the process of filtration. Okay, so let's continue our discussion now by taking a look at the second component of urine formation, which is reabsorption. So reabsorption is primarily going to occur in the lumen of the proximal convoluted tubule. So if we look at our diagram over here on our right-hand side, here's our renal corpuscle, and the filtrate is going to leave the glomerular capillaries and it's going to enter the nephron tubule. Now remember, the nephron tubule is made up of this first folded part which is near the corpuscle, which is called the proximal convoluted tubule. Then we have this looped portion, which is called the loop of Henle or Henle's loop. And then the final portion of the tubule is the distal convoluted tubule. So what we're saying here is that reabsorption is going to start, and most of reabsorption actually is going to occur in this proximal portion of the nephron tubule. We call it the proximal convoluted tubule. So here, approximately 80% of the water and varying amounts of salt and other molecules are going to be returned to the blood, and they're returned to the blood via these peritubular capillaries. Remember these traditional capillaries that we find that surround the tubular portion of the nephron. So let's see how these substances are returned. First of all, let's talk about glucose and amino acid reabsorption. Glucose and amino acid reabsorption are going to occur via the process of active transport. Now remember, active transport means that it's going to require ATP. Salt and water reabsorption, on the other hand, is going to occur via a different mechanism. So we'll break this down into steps to try to help you understand it a little bit better. The first step in salt and water reabsorption, remember salt, sodium chloride, is made up of sodium ions and chloride ions. Let's begin with the sodium ions. The sodium ions are going to be transported by active transport from the lumen of the proximal convoluted tubule back into the peritubular capillaries. Now remember that sodium ions are positively charged. Chloride ions are negatively charged. So as sodium ions are picked up and transported back from the lumen into the peritubular blood, chloride ions are going to follow them because of their charge differences. So the chloride ions will flow back into the peritubular blood as well. Now, as sodium ions and chloride ions flow back into the peritubular blood, what happens? The peritubular blood now becomes much more highly concentrated in solute because of the additional sodium and chloride ions. So what's happening? The peritubular blood immediately surrounding the tubule is becoming very high in solute concentration. It's becoming hypertonic. So what happens? We know from our previous discussions in the cardiovascular system that water always moves into the hypertonic solution. So what's going to happen is the water then is going to flow toward the solute of higher, the solution of higher solute concentration, the hypertonic solution. Where's that at? It's in the peritubular blood. So the water is going to flow out of the lumen of the tubule and into the peritubular blood. And that's how the water is reabsorbed. Okay, so let's take a look at a visual representation of reabsorption in the proximal convoluted tubule. But before we do that, let's kind of look at this simplified diagram of a nephron and go over the parts, and then we'll take each one of the parts separately and talk about what happens there. So if we can take this, we'll divide it into basically the three sections of the tubule, and this section of the tubule is the proximal convoluted tubule. This section is going to be the loop of Henle, or Henle's loop. And this section is going to represent the distal convoluted tubule. And then, again, not part of the nephron, but we're still showing it anyway. This is the collecting duct. Okay. Over here on this side, we're going to represent the glomerulus. And of course, blood enters the glomerulus through the afferent arteriole. And blood is going to exit the glomerulus here via the efferent arteriole. And of course, this is going to be the glomerulus. So this is everything that's represented. I guess I really can extend that efferent arteriole a little bit, and we could then represent the peritubular capillary here. Okay, so now what we're going to do is we're going to take a look at reabsorption as it occurs here, first of all, in the proximal convoluted tubule. Okay. Now we know what happens is that the blood enters the glomerulus through the afferent arteriole, and the hydrostatic pressure of the blood, or the blood pressure then, forces a highly dilute fluid, the filtrate, out of the glomerulus. Now, in this filtrate, there is, first of all, since this is a highly dilute filtrate, there's going to be a lot of water molecules in here. So let's add some water molecules. In addition to the water molecules in the filtrate, there is going to be a lot of nutrients like glucose. So we'll add some glucose in here. Okay, and there's going to be amino acids. There are also going to be some sodium ions. So let's add some sodium ions here, and they're going to be entering. And along with the sodium ions, there are also going to be chloride ions. So we'll add some chloride ions here. And so these are all important ions. And there are many other nutrients and substances in the filtrate as well, but we'll start by taking a look at that. Okay, so the first thing we talked about was the fact that as the filtrate passes into the proximal convoluted tubule, what's going to happen is, first, sodium ions are going to be reabsorbed in the proximal convoluted tubule. So embedded in the cells of the proximal convoluted tubule, and we're only showing one here to keep it simple and help you understand, but all of these cells in the plasma membrane of all the cells, there are going to be embedded special transporters. These special transporters are referred to as sodium glucose linked transporters type 2, SGLT2 transporters. And what the SGLT2 transporters do is they will pick up and transport sodium ions out of the lumen of the blood vessel. Excuse me, out of the lumen of the proximal convoluted tubule as the filtrate enters. Now that's how the sodium gets in. And this is an active transport process. So energy has to be used. ATP is going to be used to transport the sodium via the sodium glucose transporter type 2, the SGLT2 transporter out of the lumen of the proximal convoluted tubule and then into the cells. And then, in addition to the sodium, it's also going to transport glucose. So it's linked. It transports not only sodium, but it also transports glucose molecules. Once these are transported out, on the opposite side, on the basolateral side of the membrane, there are sodium transporters that will transport the sodium then out of the cell, and the sodium will be moved into the peritubular blood. On the other hand, the glucose will be transported via a glucose transporter into the blood as well, into the surrounding peritubular blood. Now, as the sodium ions get transported through the cells, what's going to happen? Well, chloride ions are going to want to follow. Now, there are channels for the chloride ions that are embedded in the plasma membrane of all the cells, but to keep it simple, let's move over to this cell and show one of the chloride ion channels in the cell. So here's a chloride ion channel. And so what happens now is this chloride ion is going to be transported through the channel from one side of the cell, and then there are also going to be channels on the other side of the cell. Let's put a channel to represent them over here. Okay, and the chloride ions are then going to be transported out of the cell, and they'll wind up in the peritubular blood. Okay? All right, now, next up. So this is occurring, the sodium ions are transported via an active uh, transport process. The glucose is being transported via an active transport process, and so are the chloride ions. Now, as we take the sodium and chloride ions, and for that matter, the glucose, out of the proximal convoluted tubule and transfer it into the peritubular blood, what's happening? The peritubular blood is now becoming more and more hypertonic. Okay? And what's happening to the filtrate in the proximal convoluted tubule? It's losing solute. So as it loses solute, it's becoming more and more hypotonic. So next up, let's talk about the water molecules. Okay, so now the water molecules that are in the fluid, what's happening is we now have a situation where we have a hypertonic solution out in the peritubular blood and a hypotonic solution in the proximal convoluted tubule. We know that water always moves via osmosis. It's going to move across the membrane from the hypotonic solution into the hypertonic solution. Remember, we said water always moves into a hypertonic solution. So as this becomes more and more hypertonic, that acts as a draw to draw water molecules out of the proximal convoluted tubule fluid. So let's draw some water channels in here. And by the way, these water channels that are present in the cells are called aquaporins. Okay? Because of the presence of the aquaporins, the water can move out of the proximal convoluted tubule, and it can move through the cell, and then we'll have another aquaporin on the opposite side. That's going to act as a channel to allow the water to exit the opposite side of the cell. That water is going to move into the peritubular blood. So water starts to increase in the peritubular blood. So this is how sodium ions, glucose, chloride ions, and water are retrieved or reabsorbed from the filtrate that's passing through the proximal convoluted tubule. Now, we might talk about something like amino acids as well, just to give you an example. I don't want to really focus on those too much. But when we talk about the amino acids, they are transported out via a very similar process. But this transporter right here, there's a similar transporter that can transport sodium ions and amino acids instead of glucose at the same time. And so the amino acids get transported out in a very similar fashion to what the glucose is transported out. Now, as a side right here, this SGLT2 transporter is very important clinically because there are a class of drugs that are called SGLT2 transport inhibitors. Some of these SGLT2 inhibitors are things like brands that you may have heard maybe on TV or in other places. Farxiga is a good example, an SGLT2 inhibitor. Invokana and Jardiance. These are all SGLT2 inhibitors, and they're used to help to treat diabetes. And the way they work is these transporters or these inhibitors can actually bind to and act almost like a little cork that can inhibit or block the absorption of sodium and glucose back into the peritubular blood. So when we use these SGLT2 inhibitors, let's say a person has too much glucose in their blood already, one of the ways to excrete excess glucose and get rid of excess glucose in the blood is to use these SGLT2 inhibitors that act like a cork to block the action of the SGLT2 transporter so that the glucose can't get out. And instead of getting out, what happens? The glucose remains in the filtrate in the proximal convoluted tubule. So SGLT2 inhibitors. Okay, that's what happens in the proximal convoluted tubule. Let's now move to reabsorption as it occurs in the loop of Henle. So now we're down here in the loop of Henle. And remember, the loop of Henle contains a descending limb on one side. Notice the descending limb is much thinner, and then the ascending limb has a thin part, and then it has a thick part to the ascending limb. So now what's going to happen is this glomerular filtrate is now going to move through the proximal convoluted tubule, and from the proximal convoluted tubule, it's going to move down into the loop of Henle, where it is now referred to as tubular fluid instead of filtrate. But this portion, the downward limb of the loop of Henle, remember, is the descending limb. Now the filtrate will then pass down the descending limb, and then it will pass through the loop and continue to move up the ascending limb. As the filtrate passes through the ascending limb, sodium and chloride ions are going to pass out of the tubule and begin to accumulate, and be careful about this, they accumulate in the surrounding medulla. We might say in the interstitium, the tissue of the medulla of the kidney. As a result, a hypertonic solution of tissue fluid, remember the tissue fluid is the fluid surrounding the cells of the tissue, is created in that medullary interstitium. And an osmotic gradient is going to be created. As a result, water then will flow from the descending limb into the surrounding tissues. So if you think about it, what's happening here, as the tubular fluid moves up the ascending limb, it's going to be losing sodium and chloride ions. But as it moves down the descending limb, it's going to be losing water. It only loses water on the descending side. Why? Because on the descending side, the limb is permeable to water because embedded in the cells, there are water transporting pores called aquaporins that will allow the water to pass out. These do not exist on the ascending side, so water does not exit the tubule on the ascending side of the limb. However, on the ascending side of the limb, there are ion transporters called NKCC2 transporters that we'll look at in a bit. Okay, so water exits on the descending limb side, ions exit on the ascending limb side. The water eventually will make its way back into the blood via nearby capillaries and lymph vessels. But initially, the water flows out of the descending limb into the medullary interstitium. Little or no water, as I mentioned, will leave the filtrate through the ascending limb. Why is that? Again, the ascending limb does not have aquaporins to allow for the water to get out. So we say that the ascending limb is kind of watertight, while the descending limb is water releasing. Okay, so if we take a look here at our diagram, we can see that here on the thin side, the descending loop, the water's coming out. Again, why? I know I'm sounding redundant, but that's because of the presence of aquaporins in the cells that line the tube. That allows the water to get out. And notice the water's going into the interstitium, not directly into the peritubular capillaries. As the tubular fluid now moves through the loop and into the ascending limb, here in the thick portion of the ascending limb are embedded transporters called NKCC2 transporters. These NKCC2 transporters will allow sodium and chloride ions to be transported out into the interstitium. Now, they also transport calcium, or not calcium, but potassium ions. We don't see any potassium ions out here because what happens is, at least most anyway, of the potassium ions get recovered into the ascending limb. So what's happening here? Well, we have to wait. If this is the first run, but this is occurring simultaneously, but if you think about this the first time, the water doesn't come out until the concentration of ions goes up and makes the interstitium hypertonic. Once the interstitium becomes hypertonic, water moves from the hypotonic solution into the hypertonic solution. So on this side, once the interstitium becomes hypertonic, the water now begins to flow through the aquaporins and out into the interstitium. What's important to remember here is that the ions are coming out on the upside, the ascending limb, while the water is coming out on the downside or the descending limb. This results in what is referred to as counter-current exchange. Counter-current exchange refers to the fact that the exchange of water and solutes occurs between the fluids moving in opposite directions. The water is coming out when the tubular fluid is moving down the descending limb. The ions are coming out when the fluid is moving up the ascending limb. Okay, so let's take a look at what happens with reabsorption in the loop of Henle. So here's our simplified diagram of just that section of the nephron tubule that we call the loop of Henle. So what's going to happen now is water is going to, or I guess I should say the filtrate is now going to leave the proximal convoluted tubule and travel into the loop of Henle. The filtrate, now called the tubular fluid, is going to move down the descending limb through the loop and up the ascending limb. Now, one of the things we want to mention here at this point in time, the nutrients have pretty much been removed. All of the glucose, amino acids, and other nutrients were removed in the proximal convoluted tubule. That's why this is now no longer referred to as filtrate, but now referred to as tubular fluid. So let's see what happens now as the tubular fluid moves through the loop of Henle. So we start here on this side and we follow the tubular fluid around until we reach the thick ascending limb of the loop of Henle. Remember, this is referred to as the descending limb. This is the ascending limb, and there's a portion of the ascending limb known as the thick ascending limb. Now, just like with the proximal convoluted tubule, we're going to see cells that line the thick portion of the ascending limb. I've drawn one big cell here to represent what's going on. But remember, the cells are going to line uh, the lumen on either side. So as the fluid moves up through the ascending limb, what's going to be? Well, there's going to be a lot of water in it. So we'll add some water molecules here. And in addition to water, there is also going to be sodium ions. So we'll put some sodium ions in here. And of course, there are going to be chloride ions. So it's still pretty salty. And there's even going to be some potassium ions in there. We'll just use black to represent the potassium ions. Oops. So we've also got some potassium ions. Okay. Now, embedded in the cells on this side of the loop of Henle in the ascending limb, there are transporters once again. This embedded in this cell, this transporter is called an NKCC2 transporter, because this transporter, as the fluid comes by, has an affinity for sodium ions, and what it'll do is pick up sodium ions and transport the sodium ions out of the lumen of the thick ascending limb and into the cell. In addition, it's going to pick up a potassium ion and transport the potassium ion from the lumen to the cell. Finally, it's going to pick up not one, but two chloride ions and transport the two chloride ions from the lumen to the cytoplasm of the cell. So it picks up one sodium ion, one potassium ion, and two chloride ions. That's why it's called an NKCC2 transporter. Now, what's going to happen is over here in the basolateral section, there are going to be potassium transporters that are going to transport potassium out. There will be a sodium transporter that will transport sodium out. And there will be some open channels. Let's draw some chloride channels in here. There are going to be some chloride channels here that are going to allow the chloride ions to pass out of the cell. Now, what happens to these ions once they pass out of the opposite side of the cell? Well, these ions now begin to accumulate in the interstitium of the medulla. Now, not in the peritubular blood, but in the tissue that makes up the in between the cells of the tissues that make up the medulla. So we're going to have an increase in sodium ion concentration. We're going to have an increase in chloride ion concentration. And we'll actually have an increase of other ions as well. And we'll have some increase in the potassium ion concentrations. But what will happen is there will be open channels here that will allow the potassium ions to be transported back into the cell, and then another channel here that will allow the potassium ions to be transported through the cell and back into the lumen of the thick ascending limb. So there's not much of a potassium accumulation there, but what's going to happen is the sodium and chloride ions are going to start to accumulate in the fluid around the cells that make up the tissue of the medulla. Okay, so now what happens? Well, as these ions come out, you can imagine that the interstitium is going to become what? It's going to become very hypertonic because of the loading of all these sodium and chloride ions. So now what's going to happen? Does water come out? Well, no water on this side comes out because there are no channels to allow the water to come out in the ascending limb. We say the ascending limb is kind of watertight. But in the descending limb is where we're going to find in the cells of the descending limb. I'm not going to draw the individual cells. Well, let me just put cells kind of like this in the descending limb. So embedded in these cells in the descending limb, then, there are going to be aquaporins. And these aquaporins then are going to be channels that are going to allow water to come out from the descending limb. So water molecules now are going to exit and pass out of these cells here as the water passes down, as the fluid passes down the descending limb. And so the water passes through the cells and enters the interstitium. So now this becomes more dilute, and eventually the water and the sodium and chloride ions will get transferred into the vasa recta portion of the peritubular capillaries. But first, everything gets transported into the interstitium. So what's happening here is, as we mentioned in the slides, the water exits on the descending side. Now remember, none of the ions are going to exit on this side because there are no transporters for ions in the descending limb, only for water. In the ascending limb, there are no channels to allow the water to escape, so water doesn't escape. But this transporter here, for example, the NKCC2 transporter, allows the ions to be transported out of the lumen and into the interstitium, creating a hypertonic solution. So the water escapes on one side as the fluid moves down the descending limb, and ions escape on the opposite side as the water moves out up the ascending limb. So this is what's called counter current exchange, because water's being lost as the fluid's moving down, while ions are being lost as the fluid is moving up. Now, another clinical significance of this is this NKCC2 transporter. There are some drugs that are classified as loop inhibitors. I'm sure you'll hear about loop inhibitors as you move into your clinical classes. Probably the most well-known and most commonly used of the loop inhibitors is furosemide. Also known by its brand name, Lasix. Furosemide or Lasix is an NKCC2 inhibitor. So furosemide or Lasix inhibits the ability of the NKCC2 transporter to pick up and transport the sodium chloride and potassium ions out of the lumen and into the interstitium. So the significance of that is that if you don't transport as many sodium and chloride ions out into the interstitium, this interstitium never becomes as hypertonic as it otherwise would. So, with this being less hypertonic, remember, water flows into the hypertonic solution. So this being less hypertonic, while there are channels that allow the water to flow out, it does not flow out as rapidly because there's not as steep a gradient, concentration gradient. So less water flows out and more water is retained in the loop. So that if you have someone who's retaining water, or let's say they have peripheral edema from congestive heart failure, and you want to sort of bleed off that excess water, here's a way it can be done. You can prescribe a loop inhibitor like Lasix so that not as much water is recovered from the tubular fluid in the nephrons, and therefore the individual winds up retaining the water. The water is passed to the collecting duct, and then there is more water in the urine. So these loop inhibitors are called diuretics because they increase the loss of water. This helps to then kind of, as we said, bleed off the excess water from the uh, tissues whenever we have peripheral edema. Let's move now to the distal convoluted tubule. Here, water and salt are going to be reabsorbed via a very similar mechanism to what happened in the proximal convoluted tubule. And the water and salt are going to be deposited back into now the peritubular blood, not the interstitium now, but back into the peritubular blood. So let's take a look at, excuse me, at reabsorption now. This is going to represent the distal convoluted tubule and the collecting duct. So what's going to happen now is the tubular fluid is now going to enter the collecting duct, recall, from the loop of Henle. And the fluid is going to move through the collecting duct in this direction. Okay, now in the fluid that enters the distal convoluted tubule, there are going to be what? Still some sodium ions there. So we'll add some sodium ions. And of course, there are going to be some chloride ions along with those sodium ions. And obviously then, this is still pretty highly dilute, so there is water also, water molecules in the lumen of the distal convoluted tubule. Now, for the sake of our discussion, what we're going to do is let's divide the distal convoluted tubule into two parts. And this part we're going to call the early part, and this part we're going to call the late part of the distal convoluted tubule. So here's what happens. In the early part of the distal convoluted tubule, as the fluid enters the lumen of the tubule, cells surrounding the lumen of the tubule, and let's use this cell to represent the various cells that would be lining here all along, have embedded on their luminal surface of their plasma membrane, have special transporters. These are sodium chloride. These transporters are called sodium chloride symporters. These sodium chloride symporters will attract the sodium ions and pull the sodium ions into the cell. And at the same time, they're a symporter, so they can transport more than one ion at a time. In this case, they're going to pull chloride ions into the cell as well. All right, so once the sodium and chloride ions are pulled into the cell, this is going to reduce the concentration of sodium chloride here in the tubule. Now, what happens to the sodium and chloride ions? Well, the sodium ions will travel up here and on the opposite end of the cell, on the basolateral surface of the cell, there are going to be, again, two transport proteins embedded in the cell. Now, you're familiar with these. These transport proteins are called sodium, potassium exchange pumps, which we've seen in the nervous system before. And what's going to happen is the sodium potassium exchange pump is going to pick up the sodium ion and transport the sodium ion out and into the peritubular blood. Now, while it does that, what it's going to do is it's going to pick up any potassium ions from the peritubular blood and transport the potassium ions into the cell. And this is the way substances like potassium ions get secreted from the peritubular blood back into the lumen. Eventually, these potassiums will enter channels that will allow them to be deposited back into the tubular fluid in the distal convoluted tubule. So that's how the sodium ions get out. Now, what about the chloride ions? Well, embedded in the basolateral membrane of the cell, there are chloride ion transport channels as well. So the chloride ions will now be transported through the channels, and eventually they will make their way also back into the peritubular blood and be recovered that way. Now, one of the things that we see here is here in the early distal convoluted tubule, this is still pretty watertight because these cells, notice, don't have any aquaporins in them. So there's no way for water to get out. However, whenever we move to the late end of the distal convoluted tubule here, what we find is that these cells have aquaporins embedded in their basolateral membranes. So let's draw some aquaporins in here. But the problem is these cells have the aquaporins in their basolateral membranes, but they don't have aquaporins in the luminal portion of the plasma membrane. So they're still pretty watertight. That is to say, unless there is a hormone present, and that hormone is anti-diuretic hormone. So let's say that we've seen the water be recovered in the proximal convoluted tubule. We've seen water recovery in the distal convoluted tubule. So as the water is being recovered, what's happening? The peritubular blood volume is going up. But let's say that the blood volume doesn't increase enough, and this maybe results in low blood pressure, and that creates problems for capillary exchange. So as the blood leaves the kidneys and now circulates around into the brain and passes through the hypothalamus will identify that the blood volume is too low. Well, when the blood volume is too low, the hypothalamus will then stimulate the pituitary gland to release this ADH, this antidiuretic hormone or vasopressin. When antidiuretic hormone is released, it travels through the blood and it passes out of the blood through the peritubular capillaries. And what the antidiuretic hormone does then is it stimulates these cells to add aquaporins to the luminal surface, here and here. Now, with the channels in the luminal surface plasma membrane, water molecules now have a way to be able to pass out of the distal convoluted tubule and enter the cells, and then we can follow the path where they'll pass from the cells into the peritubular blood. And again, what this will do is increase the blood volume. But this additional water is not pulled out unless the blood volume is too low. Now, I should mention here also that ADH also affects cells that line the distal collecting tubule. So here's a cell to represent the cells that line the distal collecting tubule. Now, these cells also have aquaporins embedded in their basolateral membrane. But they don't have the aquaporins embedded in their luminal plasma membrane on the luminal side. That is to say, unless ADH is present. In the presence of ADH, again, aquaporins will be added. They're not present normally, but they're added only if ADH is present. And then that allows for water molecules to be transported through the cells. But this time in the distal collecting duct, they're transported through the cells and into the interstitium. And from the interstitium, then the water gets transported into the vasa recta. So additional water can be reabsorbed in this way. So now we can also talk about a second hormone that influences water reabsorption and therefore blood volume. So again, with the scenario that the blood volume is too low because not enough water has been reabsorbed. So ADH is produced, and ADH affects the cells in the distal convoluted tubule and in the collecting duct by adding aquaporins to the luminal plasma membrane surface. This allows additional water to start to escape from the cells. A second hormone that's also involved in this is aldosterone. Aldosterone is produced by the adrenal gland. Aldosterone enters the blood, and when aldosterone makes its way to these cells in the distal convoluted tubule, here's what the aldosterone does. Already embedded in the cells in the distal convoluted tubule, there are sodium-potassium exchange pumps. These sodium-potassium exchange pumps do their job. They exchange sodium and potassium between the collecting duct and the cells that line the collecting duct. And they effectively pull more sodium out of the collecting duct. But under the influence of aldosterone, additional sodium-potassium exchange pumps are added. And what this does is allows a quicker exchange of sodium and potassium ions. So sodium ions then are transported out now of the collecting duct at a faster pace. And of course, there are sodium channels over here on the other side, and the sodium gets transported out of the cell more quickly. What does that do? That's going to increase the amount of solute again in the interstitium. And that's going to cause the interstitium to become more hypertonic. The more hypertonic the interstitium is, getting back to this water flow, the faster the water is going to flow through the channels and be removed from the collecting duct. And eventually, the water will, from the interstitium, be returned to the blood via the vasa recta. So the idea here is not only do we want to increase the channels to allow the water to flow out, but we want the water to flow out more quickly, and that's where adding the sodium-potassium exchange pumps comes into play. And that increases the uh, hypotonic nature or hypertonic nature, excuse me, of the interstitium. And so the water not only flows out, but it flows out more quickly to reduce the amount of water in the urine and increase the amount of water in the peritubular blood. So that helps to control water balance. These are the two hormones that are involved in water balance. ADH, which works in the distal convoluted tubule, and aldosterone, which works in the collecting duct. And ADH actually works in the collecting duct as well. So ADH, once again, in the distal convoluted tubule, late part, and in the collecting duct, and aldosterone working in the collecting duct to increase the sodium-potassium exchange pumps and help with water balance. All right, that takes us to the third process of urine formation, which is secretion. Secretion, we could actually say, is reabsorption reversed. It's the opposite of reabsorption. So in reabsorption, we're trying to pull nutrients and important ions back out of the tubular fluid and put them back into the blood. In secretion, what we're trying to do is take substances such as hydrogen ions, potassium ions, creatine, which would be a waste product, and we're trying to move them from the blood of the peritubular capillaries and put them in to the glomerular filtrate. These were maybe excess, for example, hydrogen ions or excess creatine that were not filtered out in the renal corpuscle, but still need to be removed from the blood. So these additional substances can be secreted back into the tubular fluid. This primarily occurs in the proximal convoluted tubule as well. Now, the importance of secretion is several-fold. First, again, this helps us to dispose of substances, helps the body to dispose of substances that are not already in the filtrate. Maybe excess hydrogen ions, potassium ions, creatine, or even things like certain drugs that we may take. They'll eventually pass out through the urine through this process of secretion. And that's why we have urine testing and why it works, because certain drugs, as they circulate around, will be filtered out, not filtered out, but will be secreted out of the blood into the tubular fluid and eventually can be found in the urine. Another importance of secretion is the elimination of undesirable substances that have been reabsorbed by passive processes. So maybe this doesn't happen very much, but maybe the waste products, other waste products like urea and uric acid were in the filtrate, but they got reabsorbed by passive processes um, from the tubular fluid. Well, we don't want to reabsorb those. We don't want to retain them in the body. So they can then be resecreted back into the tubular fluid. Also, another importance is to eliminate any excess potassium ions. So you recall that we said on the previous slide, for example, we were looking at the loop of Henle that we didn't see potassium ions because they could be resorbed. Well, if there are too many potassium ions, we still maybe want to get rid of them in the urine. So by the process of secretion, excess potassium ions can be eliminated in the urine. And of course, another importance of secretion is the control of blood pH, right? Because pH is a function of the relative concentration of hydrogen and hydroxide ions. If the number of hydrogen ions is high in the blood, the pH is much lower. We want to keep the pH of the blood right around 7.3, right? As we discussed in the past. Well, if the pH is a little bit too low, that means there's still too many hydrogen ions in the blood. And if we haven't if we haven't filtered out enough hydrogen ions, then through the process of secretion, we can add additional hydrogen ions to the urine to get them out of the body and therefore bring the pH back down. So this is the importance of secretion. All right. Now, next, let's take a look at how the nitrogenous waste products are excreted via the formation of urine. There are three important nitrogenous waste products that are found in the blood. We've talked about one, actually all of them already: creatine, urea, and uric acid. Let's take a look at urea first. This is formed in the liver when the amino acids are used for energy. You might recall from nutrition, if amino acids are going to be used for energy, what has to happen? The amine group has to be removed. And when the amine group is removed, it then is attached to other substances, and it's a convoluted process, but it results in the formation of this waste product called urea. So urea is an end product of protein breakdown. Our next nitrogenous waste product is uric acid. Uric acid is released when nucleic acids are metabolized in the body. Now recall that nucleic acids are used for the formation of, uh, uh, I should say the nucleic acids are DNA and RNA, but they can be metabolized from the foods that we eat. And whenever they are and they're broken down, one of the byproducts of that breakdown is uric acid. And uric acid has to be eliminated from the body. It's eliminated through the urine. And then finally, there's that creatine. Recall that creatine is produced as a result of creatine phosphate breakdown in muscles. So when our muscles are active, creatine phosphate can be used to resupply ATP, to regenerate ATP in muscles by transferring a phosphate group to an ADP. Well, what happens to the creatine? Well, the creatine is released into the blood and then has to be removed by the kidneys. Because these wastes are large and cannot be actively transported, with the exception of creatine, they tend to remain in the filtrate once they're pushed out and are found in high concentrations in urine. All right, next up, let's talk about water balance. So recall that the job of the kidneys is to control the composition and concentration of the plasma and the interstitial fluid. Concentration deals with how much water is in the blood. So we want to have the right amount of water so we have good capillary exchange. So what controls the water balance, the appropriate concentration of water? Well, the rate of reabsorption in the distal convoluted tubule and the collecting duct is controlled by two hormones. The first of these hormones is called antidiuretic hormone. Antidiuretic hormone is produced by the hypothalamus and it's secreted by the posterior lobe of the pituitary gland. Antidiuretic hormone will increase the permeability of cells in the distal convoluted tubule and the collecting duct. The permeability of cells to what? To water. And we're going to see, I'll show you a little diagram a little bit later, that the increase in permeability is due to the addition of water channels, aquaporins, into the cells of the distal convoluted tubule and into cells of the collecting duct. The result is an increased flow of water back into the peritubular blood. So the scenario is this. Let's say that the filtrate comes out, the water's reabsorbed. Let's focus on the water here since we're talking about water balance. The water gets reabsorbed, but by the time we reach the distal convoluted tubule and collecting duct, let's say that enough water has not been reabsorbed into the peritubular blood to maintain the proper blood volume. And additional water has to be pulled back into the blood. Well, antidiuretic hormone will be released by the pituitary gland, and it will travel to the distal convoluted tubule where it will bind with receptors on cells and in the collecting ducts and stimulate those cells to add aquaporins to their plasma membrane. This will allow additional water to move back into the peritubular blood to get the balance where it needs to be. If the peritubular blood already has enough water in it, then the antidiuretic hormone will not be secreted, the aquaporins will not be embedded in the membrane, and additional water will not be recovered. It'll pass out with the urine. The second hormone that is involved in water balance is aldosterone. Aldosterone is produced by the adrenal gland. It works primarily in the collecting duct. It has three important effects on the cells of the collecting duct. First, it stimulates the reabsorption of sodium and chloride ions from the collecting duct. It does this by adding sodium and chloride transporters into the plasma membrane of the cell. With additional sodium and chloride ion transporters, the cells are able to pull more sodium and chloride ions back out of the urine and put them back into the peritubular blood. Now, this is going to stimulate also the reabsorption of water, since water molecules are going to follow those sodium ions in the process of osmosis. Third, it's going to stimulate the secretion of potassium from the blood into the fluid in the collecting duct. So potassium transporters, it'll stimulate the potassium transporters to be embedded in the cells so that they'll pull potassium from the blood and put it back into the urine to secrete excess potassium if potassium levels in the blood are too high. Okay, let's kind of start to wrap everything up here by taking a look at the other component of, or components of the urinary system, and that's the urinary tract. Remember, the urinary tract is composed of the ureters, the urinary bladder, and the urethra. Let's take a look at the ureters first. These are fibromuscular tubes that conduct urine from the kidneys to the urinary bladder. They're approximately 25 centimeters long in the human body, and like the kidneys, they are also located retroperitoneal, outside the peritoneum. The ureters will enter the posterolateral wall of the base of the bladder. And you'll see when you take a look at, if you haven't already, you take a look at the urogenital dissection of the cat, you can see very well a very similar entry location for the ureters in the cat bladder. Since the ureters are fibromuscular, they contain a lot of smooth muscle tissue and therefore are capable of carrying out peristalsis. Wave-like contractions of the smooth muscle tissue will help to move the urine from the kidneys to the bladder. Next up, we take a look at the urinary bladder itself. The urinary bladder is a smooth, collapsible muscular sac. Its job is to temporarily store urine and then release the urine at the appropriate time to the outside. Here we can see a diagram of the urinary bladder and see the positioning of the ureters. And notice down here the openings to the ureters, postero and lateral on either side. Our next, we'll take a look at some landmarks in the urinary bladder. The first one is the trigon. The trigon actually consists of three openings. Two of the openings comes from the ureters, and one opening comes from the urethra. So we can see down here at the bottom, we can see here this right uteral orifice, and here we can see the left uteral orifice. So these two openings, and in addition, the urethral opening here kind of makes a triangle, and this refers to the area at the base of the bladder referred to as the trigon. Next, we look at the bladder wall itself. The bladder wall is composed of three layers of smooth muscle. The smooth muscle is referred to as the detrusor muscle. In addition, there is a mucosal layer. That's the inner layer of the bladder up against the lumen or the interior of the bladder. The mucosa is made up of transitional epithelium, and you may recall from AMP1 that transitional epithelium has cells that are kind of dome-like, and they're kind of folded over top of one another, so that when the bladder expands as it fills up with urine, the cells can kind of unwrap or unfold from one another, and that will allow the bladder to expand. The walls of the bladder are kind of thick and folded in an empty bladder because those cells are all kind of folded over top of one another. But the bladder can expand significantly, and again, that's due to the transitional epithelium without increasing very much internal pressure. So it can fill up with urine, and you don't have the pressure to give you the urge to urinate up until a certain point. Next up, we take a look at the urethra. The urethra is a thin-walled tube that carries urine from the bladder to the outside of the body, and again, this is going to occur via peristalsis. The release of urine is actually controlled, though, by two sphincter muscles. Remember, sphincter muscles are circular pieces of muscle that kind of form valves to control the release of things from one part to another, like what we saw in the digestive system with the cardioesophageal sphincter and the pyloric sphincter. The first of these two sphincters is referred to as the internal urethral sphincter. Now, the internal urethral sphincter is involuntary. That means you don't have any control over it. The second sphincter is called the external urethral sphincter. The external urethral sphincter is made up of skeletal muscle tissue, so it is voluntary. You can control the opening and closing of the external urethral sphincter. So before we get on to gender differences, what happens then is once the bladder fills up to a certain point, let's say 300 to 350 milliliters, it'll start to put pressure on the internal urethral sphincter, and the bladder will have been sufficiently stretched to have an autonomic reflex, and it'll start to contract, putting additional pressure on the internal urethral sphincter. Spontaneously, the internal urethral sphincter will open up. You have no control over that. But when it does open, you feel the urge to urinate. But you don't urinate until you open, and this gives us time to get to an appropriate place. You don't urinate until the external urethral sphincter opens up and allows the urine to pass to the outside. Okay, let's take a look at some gender differences between the urethra of males and females. The length in females, the urethra is only about 3 to 4 centimeters, and that's the equivalent of about an inch. In males, because the urethra passes through the penis, it tends to be longer at about 20 centimeters on average, and that's somewhere around 8 inches. Location-wise, in females, the urethra runs along the wall of the vagina. And again, this is very similar to what you may have seen in the cat dissection. If you haven't seen that yet, look for it and see where the urethra is running right behind the vagina. In males, the urethra it runs directly through the prostate, and it does in cats as well, as you will see in the dissection video if you haven't looked at that. And then it runs through the penis. The function of the urethra in females is only to carry urine, but in males, it is going to carry both urine and sperm cells. Here's just a diagram indicating those gender differences and some of the different components. So here in the female, we can see here's the internal urethral sphincter, that first valve, and the external urethral sphincter, the second valve. So when the muscle, the detrusor muscle, the urinary bladder contracts, it creates pressure on the internal urethral sphincter. The sphincter opens up, but we control the external urethral sphincter, and until we decide that that sphincter will open up, um, the urine will not pass to the outside. This is a very short distance in females. In males, there's a much longer distance. So here's our internal urethral sphincter, okay, and our external urethral sphincter is going to be a little bit lower here. I don't think it's shown in the diagram here, but then we can see the length of the urethra running all the way through the penis versus just the short distance that there is from the bladder out to the body wall. All right, next up, we'll talk about micturition. Micturition is another term that we use to mean urination. Micturition is controlled by two sphincter muscles, as we mentioned. These sphincter muscles are called the internal and external uh, uh, sphincter muscles. They go by other names as well, and that's why this slide is here. Another name for the internal urethral sphincter is the sphincter vesicae. This is located near the base of the bladder. Okay, so if you think about it, it's near the internal organs, so that's where it gets vesicae from. It's composed, as I mentioned, again, of smooth muscle tissue, and that smooth muscle tissue is under control of the autonomic nervous system, so it's under involuntary control. So as I mentioned before, when approximately 300 milliliters of urine has accumulated in the bladder, a parasympathetic reflex. Remember, the parasympathetic nervous system is a branch of the autonomic nervous system, is initiated, and that causes the sphincter to open and allow the urine to flow past it. But the urine won't exit the body until the external urethral sphincter opens up, and the external urethral sphincter goes by another name. It's called the sphincter urethrae. It's located approximately 1 to 3 centimeters inferior to the sphincter vesicae. It's composed of skeletal muscle tissue, as I mentioned, and it is under voluntary control. So the presence of urine in the urethra stimulates the urge to micturate once that sphincter vesicae opens up. But since this valve is under voluntary control, micturition or urination will not occur until we choose to relax that muscle. So both sphincters have to be relaxed in order for urination to occur. And this is just a diagram, again, going over those two sphincter muscles. All right, so how does our urinary system change as we age? Well, the control of the voluntary urethral sphincter actually doesn't start until about the age of 18 months. So you don't want to try to potty train children until they're somewhere around 18 months, because they're just not going to have voluntary control over that external urethral sphincter. So once that internal urethral sphincter opens up, they're going to urinate. Okay. But after they hit 18 months, now they'll start to gain some control over it, and they'll be able to be potty trained, learn to control the passage of urine until they can get to a bathroom. Changes in the size and functioning of the kidneys will actually begin at about the age of 30. After the age of 30, there's a gradual reduction in the size of the kidneys. This gradual reduction is due to a reduced blood flow into the kidneys after the age of 30, and to a decrease in the number of functional nephrons. So after we hit the age of 30, the number of nephrons begins to decrease in the kidneys, and this reduces the kidney size. As a result of this, with less nephrons, there's going to be less reabsorption and secretion. In addition, there's going to be, as we age, a diminished ability to filter and cleanse the blood because, again, of the decrease in the number of functional nephrons. We'll also produce less aldosterone and less antidiuretic hormone. And if we produce less aldosterone and less antidiuretic hormone, while as we age, you would think that we would urinate less often, clinically, what do we see? Elderly people urinate more often. The reason for this, they are not producing as much aldosterone or antidiuretic hormone, so they're not pulling as much water back out into the peritubular blood, so they tend to produce a higher volume of urine than otherwise they would. So the ability to control blood volume and blood pressure is going to be reduced as we age. And this is one of the reasons why we have a kind of a natural creeping up of blood pressure as we age. In addition, as we age, the bladder is going to decrease in size, and that's another reason for more frequent urination. While we may be urinating more frequently, maybe we're not passing as much urine. Because the bladder is getting smaller, it can't hold as much urine as well. Control of the urethral sphincters and micturition may also be lost with age. This is in particular something that happens in women because of pregnancy, and the pressure that's created on the bladder kind of creates stresses that weaken the muscles, and then as women age, they tend to have problems with leakage or dripping. Okay, so that's the end of our second screencast on the urinary system. Next up will be the reproductive system, our last system that we'll look at this semester.